Welcome back to G Fun Facts Online. Today we are going to completely rethink how we measure time. Not on a watch or a wall clock, but inside our own bodies. And specifically how that internal time could be the key to unlocking the next revolution in cancer treatment. We are talking about chronotherapy. Now usually, when we think about cancer treatment, we think about the what. What drug is being used? What is the dosage? What are the genetic markers of the tumor? But for decades, we have largely ignored the when. In a standard oncology ward, time is logistical. It is a calendar on the wall. Chemotherapy happens every 21 days. Radiation happens Monday through Friday between 9 and 5. A dose of a toxic drug given at 9 in the morning is assumed to be biologically identical to that same dose given at 5 in the evening. But recent science is telling us that this is a dangerous oversimplification, because beneath that rigid man-made hospital schedule lies a much older evolutionary timekeeper, the circadian rhythm. Every single cell in your body, from the neurons in your brain to the cells in your liver, has a 24-hour cycle. They are not static, they are ticking. And the emerging field of chronotherapy suggests that syncing cancer treatment with these internal clocks can not only drastically reduce side effects, but also significantly improve survival rates. It is a shift from a static model of medicine to a dynamic one. So let's break down the science, the evidence, and the future of this time-based medicine. To understand why timing matters, we have to look under the hood at the machinery itself. The circadian rhythm isn't just about sleep. It is a fundamental property of life on Earth, evolved to anticipate the rising and setting of the sun. It starts in the brain. Deep in the hypothalamus, there is a tiny cluster of about 20,000 neurons called the suprachiasmatic nucleus, or SCN. This is the master clock. It gets direct input from your eyes about light and dark, and it synchronizes the body to the outside world. But here's the crucial part. The SCN is just the conductor. It sends signals to peripheral clocks located in virtually every tissue of your body. Your liver has a clock. Your heart has a clock. Your immune system has a clock. These aren't just metaphors. They are molecular realities. At the cellular level, this ticking is driven by a feedback loop of proteins. Two primary proteins, creatively named clock and BMAL1, bind together during the day to activate thousands of genes. As the day goes on, their own repressors, proteins called PER and CRY, accumulate and eventually shut them down. This cycle takes about 24 hours. Why does this matter for cancer? Because these clock genes regulate up to 50% of the human genome. They control the enzymes that metabolize drugs in your liver. They control the checkpoints that decide if a cell divides. They control the repair crews that fix DNA damage. So your body is biologically a completely different machine at 10 a.m. than it is at 10 p.m. Cancer cells, interestingly, often have broken or rewired clocks. They try to decouple themselves from this system, to divide uncontrollably, but they often still have residual rhythms, or at least they are subject to the host's rhythms, like your cortisol levels. This creates a therapeutic window. If we can attack when the cancer is awake but the healthy cells are sleeping, we might achieve the holy grail of oncology: maximum kill with minimum collateral damage. Let's look at the pharmacology of time. There are two pillars here. First, how your body affects the drug, which changes by the hour. Take the liver. It is the body's detox factory, but it works in shifts. The activity of CYP enzymes, which break down about half of all drugs, can vary several-fold throughout the day. If a chemotherapy drug is toxic to the liver, giving it when those enzymes are at peak activity might allow the body to neutralize the excess quickly, reducing side effects. Second, we have how the drug affects the body. Many chemo drugs target dividing cells. In healthy tissues like your gut lining or bone marrow, cell division is tightly gated by the clock, often peaking at night to avoid UV damage during the day. If healthy gut cells are dividing at 2 a.m., and you pump in a toxic drug at that exact moment, you are going to cause severe damage. But if you wait until 2 p.m., when those cells are resting, that same drug might be harmless to the gut while still killing the tumor. This isn't just theoretical. We have decades of evidence, starting with colorectal cancer. This has been the flagship for chronotherapy, largely thanks to pioneers like Dr. Francis L. Vicks. The standard treatment for metastatic colorectal cancer is a cocktail called Folfox, which includes a drug called oxaliplatin. In standard care, these drugs are just infused whenever, but in chronomodulated trials, they timed it precisely. They delivered the peak dose of oxaliplatin at 4 p.m. and the peak dose of another drug, 5-FU, at 4 a.m. The results from early trials were stunning. The timed schedule reduced severe mouth sores by 5-fold and nerve damage by half. Because the patients could tolerate the drug better, doctors could safely increase the dosage, leading to better tumor shrinkage. However, science is rarely a straight line. Later, larger trials showed mixed results on overall survival. When they dug into the data, they found a fascinating reason: biological sex. Men benefited significantly from that specific schedule. Women did not. It turns out, the female cellular clock may run slightly earlier or respond differently than the male clock. It wasn't that chronotherapy failed. It was that the one-size-fits-all timing failed. We needed personalized timing. Let's move to brain cancer. Glioblastoma is incredibly aggressive, and the standard drug is temozolomide. Researchers analyzed data from patients and found something remarkable. Patients who took their pill in the morning survived significantly longer, about 17 months on average, compared to those who took it in the evening, who averaged about 13.5 months. That is a massive difference just from changing the time of day. In patients with a specific genetic marker, morning dosing extended survival by roughly 6 months. The theory is that the DNA repair crews that fix the damage the drug tries to cause are less active in the morning, making the drug more lethal to the tumor. Perhaps the most exciting frontier is immunotherapy. These are drugs that wake up the immune system to fight cancer. But the immune system has a very strong daily rhythm. Cortisol, which suppresses the immune system, is high in the morning. But the trafficking of T cells and the efficiency of antigen presentation is also very active during the day. A major study published in The Lancet Oncology looked at stage 4 melanoma patients. They found that patients who received their infusions in the late afternoon after 4:30 p.m. had a significantly higher risk of death compared to those treated in the morning. It appears that giving an immune booster when the immune system is naturally winding down for the evening is far less effective. It is like trying to start a car when the engine is cold. So if the evidence is this strong, why isn't your local hospital doing this? Why isn't every infusion timed to the minute? The challenge is variability. We are not all built the same. We have morning larks and night owls. A lark might have a fast internal clock, say 23.8 hours. An owl might have a slow clock, maybe 24.5 hours. If a clinical trial treats everyone at 4 p.m., for a lark that might be biological evening, but for an owl, it is biological afternoon. By treating everyone at the same wall clock time, we are inadvertently treating many patients at the wrong biological time, washing out the results. To fix this, we need technology. We need to stop looking at the clock on the wall and start looking at the clock in the blood. This is where personalized chronotherapy comes in. We are seeing breakthroughs like time signature, a blood test developed at Northwestern University. By analyzing just 40 genes from a single blood draw, a machine learning algorithm can predict your precise internal body time to within an hour and a half. So a doctor could say, your internal biological noon is actually 2 p.m. and adjust your treatment accordingly. Then there are wearables. We can use smartwatches to monitor body temperature and rest activity cycles to map a patient's rhythm without invasive tests. And crucially, we need the hardware to deliver the drugs. Most infusion centers are only open 9 to 5. If your optimal dose time is 4 a.m., a standard clinic can't help you. This has driven the invention of smart pumps, like the M-Lodi pump. This is a portable device a patient can wear in a fanny pack at home. It is programmable to ramp up the drug flow while the patient sleeps, hitting that peak at 4 a.m. automatically. This solves the logistical nightmare and allows for precise, sinusoidal delivery rates that match biology. We are moving toward a future of precision chronomedicine. Imagine a world where chrono triage is standard. Morning slots at the clinic are reserved for patients with specific chronotypes or cancers like melanoma that respond best to morning treatment. Imagine a closed-loop system where a wearable sensor talks to an implanted drug pump, releasing chemotherapy at the exact millisecond your healthy cells are most protected. This isn't science fiction. The pieces are already here. Chronotherapy challenges us to rethink our relationship with time and medicine. We have spent billions focusing on the sharpness of the sword, developing more potent drugs. But chronotherapy teaches us that the timing of the swing is just as important. By syncing our treatments with the ancient, rhythmic pulse of our biology, we can turn the body's own clock into a powerful ally. The future of cancer care is not just personalized, it is synchronized. That is it for today's deep dive. Thanks for listening to G Fun Facts Online.